The TEC family kinases contain a number of regulatory regions outside of the catalytic kinase domain, the plextrin homology, TEC homology or PHTH domain, a proline-rich region or PRR, a Src homology 3 or SH3 domain, Src homology 2 or SH2 domain, and a linker that connects the end of the SH2 domain to the beginning of the kinase domain. The current model for the autoinhibited structure of ITK is shown here and is based on a crystal structure of the SH3-SH2 kinase portion of the molecule and solution studies of the full-length protein using NMR spectroscopy and hydrogen deuterium exchange mass spectrometry. The SH3 and SH2 domains assemble onto the back of the kinase domain opposite the active site, with the SH2 kinase linker sandwiched between these regulatory domains and the kinase domain. The PHTH domain sits on the opposite face of the kinase domain and sterically occludes the activation loop. For the SH3, SH2, and PHTH domains, each of the ligand binding sites is either occupied or masked in this autoinhibited form. Taking a closer look at each of the regulatory interactions, we can zoom in on tyrosine 511 in the ITK activation loop. Phosphorylation of this regulatory tyrosine by the upstream kinase, LCK, in T cells is required for activation of ITK catalytic activity. The structure shown is the inactive state of the kinase where tyrosine 511 is not accessible due to the conformation of the activation loop and binding of the PHTH domain. Next, the long linker between the ITK-PHTH domain and the SH3 domain contains a proline-rich region that matches the typical SH3 domain ligand sequence. Solution data show that the proline-rich region binds to the adjacent SH3 domain in an intramolecular fashion, and in the context of full-length ITK, the PRR displaces the SH3 domain from its autoinhibitory interaction with the SH2 kinase linker. In the fully autoinhibited form, the ITK-SH3 domain contacts the SH2 kinase linker region in much the same manner as the previously characterized Src family kinases. The autophosphorylation site, tyrosine 180, sits in the ITK-SH3 binding pocket, and phosphorylation of this tyrosine likely leads to changes in ligand binding affinity for the ITK-SH3 domain. Since the mid-1990s, when the ITK sequence was identified, it has been clear that ITK lacks the C-terminal regulatory tail present in the Src family. In Src, the regulatory tail contains a conserved tyrosine that serves a crucial regulatory role. When phosphorylated, the tail phosphotyrosine binds intramolecularly to the SH2 domain to stabilize the autoinhibited conformation. In the absence of this interaction, it has been unclear how ITK autoinhibition is mediated in this region of the protein. We have identified a conserved salt bridge between arginine 265 and glutamate 617 that mimics the Src regulatory interaction. Glutamate 617 at the very end of the kinase domain locks the SH2 domain into its autoinhibitory pose, and mutation of glutamate 617 or binding of a phosphotyrosine containing ligand to the SH2 domain will activate the kinase. Now we will consider how autoinhibited ITK is integrated into the larger signaling complex downstream of the T-cell receptor. T-cell receptor engagement leads to activation of two kinases, LCK and ZAP70. LCK phosphorylates ITAM motifs associated with the T-cell receptor, which creates docking sites for ZAP70. LCK then phosphorylates ZAP70, which goes on to phosphorylate tyrosines on the adapter proteins LAT and SLP76. These phosphorylation sites create binding platforms for numerous signaling molecules, shown here only briefly for clarity. For activation of ITK, the phosphotyrosine at position 145 in SLP76 is crucial. It binds to the ITK-SH2 domain, disrupting the autoinhibitory contact between ITK's SH2 and the end of the ITK kinase domain. ITK opens further with the SH3 domain binding to proline-rich regions in the adapter complex, and at the same time, the ITK-PHTH domain engages its ligand, IP3, which has been created by the activity of IP3 kinase at the membrane. In addition to the conformational changes accompanying the shift from autoinhibited ITK to active kinase, LCK phosphorylates the activation loop tyrosine, tyrosine 511, to fully stabilize the kinase domain in its active state. Once fully active, ITK can phosphorylate its own SH3 domain and is thought to phosphorylate another tyrosine on SLP76, tyrosine 173. The ITK SLP76 LAT complex is poised to receive the substrate of ITK, phospholipase C-gamma-1 or PLC-gamma-1. PLC-gamma-1 adopts its own autoinhibitory structure prior to activation by ITK. Shown here is just the central portion of the phospholipase, the specific array, which contains a split pH domain, 2 SH2 domains, and an SH3 domain. Embedded between these domains is tyrosine 783, the target of the ITK kinase domain. The SH2 domains of PLC gamma 1 bind to two different phosphotyrosines on the lat-SLP76 complex. The first SH2 domain of PLC gamma 1 binds to phosphotyrosine 132 on lat, and the second SH2 domain binds to phosphotyrosine 173 on SLP76. These interactions serve to open the autoinhibited PLC gamma 1 structure to reveal the ITK substrate tyrosine 783, as well as the docking site on the second PLC gamma 1 SH2 domain that ensures specificity in the phosphotransfer reaction. Biochemical evidence suggests that once tyrosine 783 on PLC gamma 1 is phosphorylated, it will compete with tyrosine 173 on SLP76 for binding to the second PLC gamma 1 SH2 domain. The intramolecular phosphotyrosine 783 SH2 interaction within PLC gamma 1 will promote release of the phospholipase from the ITK SLP76 lat complex. The ITK mediated catalytic cycle continues with another molecule PLC gamma 1 coming into the ITK SLP76 lat complex, engaging in specific SH2 mediated interactions both via the phosphotyrosines of lat and SLP76 and through the non-phosphotyrosine mediated docking interaction between the second PLC gamma 1 SH2 domain and the ITK kinase domain. Again, ITK phosphorylates tyrosine 783 in PLC gamma 1, leading to release of another molecule of activated phospholipase. The pool of activated PLC gamma 1 mediates cleavage of PIP2 to produce IP3 and diacylglycerol, which are important second messengers that initiate calcium flux and activate the RAS MAP kinase pathways.